The possibility that senolytic drugs, originally developed to target aging and dysfunctional zombie cells, could slow or even reverse the progression of Alzheimer's disease and other neurodegenerative conditions, is one of the most provocative ideas emerging in longevity and neuroscience research today. While much of the early work with senolytics has focused on physical decline in tissues like the joints, spine, and cardiovascular system, recent studies suggest that cellular senescence also plays a central and underappreciated role in the aging brain. This opens a compelling new window into how we might treat diseases long considered irreversible. The traditional view of Alzheimer's disease has centered on amyloid beta plaques and tau protein tangles, misfolded proteins that accumulate in the brain and disrupt communication between neurons. However, treatments that directly target these proteins have yielded limited cognitive benefits in clinical trials. This has pushed researchers to explore upstream mechanisms that might drive or exacerbate neurodegeneration, and one of the most promising culprits is cellular senescence, specifically in non-neuronal brain cells like astrocytes, microglia, and oligodendrocyte progenitor cells. In a healthy brain, these glial cells support neurons, maintain the blood-brain barrier, and regulate inflammation. But with age and cumulative stress, some of these cells enter a senescent state. Though they stop dividing, they remain metabolically active and begin to secrete a toxic mix of inflammatory cytokines, chemokines, and proteases, hallmarks of the senescence-associated secretory phenotype, SASP. This inflammatory microenvironment not only damages nearby neurons but may also reduce synaptic plasticity, impair memory formation, and accelerate neuronal loss. Over time, it contributes to the hallmarks of dementia: confusion, memory impairment, and cognitive decline. What makes this discovery so significant is that these senescent glial cells appear to accumulate in regions most vulnerable to Alzheimer's, including the hippocampus and prefrontal cortex. In animal models of Alzheimer's disease, researchers have identified increased markers of cellular senescence, such as P16-INK4A, P21, and DNA damage response proteins in these brain regions. Even more compelling, studies using senolytic drugs like dasatinib and quercetin have shown that clearing these senescent cells reduces neuroinflammation, improves synaptic function, and leads to measurable cognitive improvements in mice. In one study, senolytic treatment in aged mice with Alzheimer-like pathology led to improved maze navigation, reduced beta-amyloid burden, and increased neuronal survival. This suggests that senolytics do not need to target the neurons themselves, which are typically not senescent, but instead can rejuvenate the neural environment, making it more conducive to healthy brain function and less prone to inflammatory damage. By removing the bad neighbors, senolytics may allow healthy brain cells to communicate more effectively, regenerate where possible, and resist the chronic stress that leads to degeneration. Moreover, neurodegenerative diseases beyond Alzheimer's, including Parkinson's disease, frontotemporal dementia, and even multiple sclerosis, may involve cellular senescence in the supporting glial populations. In Parkinson's, for example, senescent astrocytes and microglia contribute to the loss of dopamine-producing neurons in the substantia nigra. In multiple sclerosis, senescent oligodendrocyte progenitors lose their capacity to remyelinate damaged neurons. In both cases, removing these dysfunctional support cells has shown promise in preclinical models, potentially halting or even partially reversing disease progression. Of course, translating these findings from rodents to humans is a major leap. The human brain is more complex, and delivering senolytic drugs past the blood-brain barrier remains a technical challenge. Not all senolytics penetrate the central nervous system efficiently, which may limit their effectiveness. However, new molecules are being designed with better brain bioavailability, and nanoparticle delivery systems or intranasal administration routes are under investigation to overcome this barrier. Another concern is safety. While senolytics are designed to eliminate only cells with specific survival pathway activation, common in senescent cells, the risk of off-target effects must be carefully managed, especially in the brain, where precision is paramount. Yet early-phase human trials are already underway, testing the effects of senolytics like Physidin on cognitive decline, frailty, and inflammation in older adults. These trials will provide vital data on tolerability and potential neuroprotective effects. Perhaps most importantly, the senolytic strategy aligns with a systems-based approach to neurodegeneration. Instead of focusing narrowly on protein aggregates or neurotransmitters, this approach acknowledges that aging itself, through mechanisms like inflammation, mitochondrial decline, and senescence, is the primary risk factor for dementia. If we can slow the biological aging of brain tissues, we may not just delay Alzheimer's, we may prevent or transform the trajectory of many neurodegenerative diseases. In conclusion, the application of senolytic drugs to brain aging and dementia is still in its early stages, but the foundational science is strong, and the logic is sound. By clearing zombie cells in the brain, we may not only reduce the neuroinflammatory noise that contributes to cognitive decline, but also help the brain restore its capacity for resilience and repair. While more clinical data are needed, this represents a deeply hopeful direction for the future of neurodegenerative disease treatment, one that targets not just the symptoms, but the aging processes at their root.